All right, good morning. Thanks, Con, for the invitation. I always love coming back down here to California. I did my PhD work here at UC Davis and worked on irrigation management. So here I am, and we're going to talk about water relations. And what do I got? 40 minutes? We'll see how that goes. I found this quote at the beginning of this Woody Plant Physiology book by Kramer and Kozlowski, and I replaced all the places where they wrote trees with vines, and I replaced all the places where they wrote horticulturist with viticulturist, but the idea is the same. A lot of the times there's a sort of a question about this kind of applied nature that we're thinking about how do we manage the vines in the vineyard, and then there's a question about what do we know about grapevines? And I always like to tell people, I'm trying to do two things at the same time. I'm trying to figure out how grapevines grow, and I'm trying to figure out how to grow grapevines. And I think both are tied very closely together. And so that's exactly what this sort of quote says. The two objectives are very closely related than generally supposed, because in order to grow vines efficiently, one must understand basic physiological processes that control growth. And the greatest overall progress is when we marry the two. So it's incredibly important to understand the physiology before you can start to manipulate and manage vines. Although I think we're all kind of there, I just wanted to sort of start us off. And this being a water management course, before we talk about managing water in the vineyard, I wanted to go over something about water because it's kind of a special molecule. And I had this guy from Silverado was like, "You're not going to talk about hydrogen bonds, are you?" And I said, "Yeah, I am." So hydrogen bonds are right here. Water has really unique chemical and physical properties, and most of them are derived from the structure, and water is a highly polar molecule, and the polarity and the molecular shape leads to this hydrogen bonding among the molecules. And water is a liquid at standard temperature and pressure. And in contrast to other molecules that are the same size as water, that are the same size as water. Let me try and function here. Something like ammonia or methane, carbon dioxide, all of those molecules are gases at standard temperature and pressure. And so water's size and polarity make it a really great solvent. So it serves as a really nice medium for biochemical reactions. Like, anyone think of a really important biochemical reaction in grapevines that water is like a main part of? So what? You're not allowed, no speakers. Photosynthesis, right? And water is also a really good solvent and involved in solute transport. So what's really important solute transport in grapevines? How about sugar coming into the fruit, right? So the extensiveness of the hydrogen bonding also confers really unusual thermal properties to water. Has a really high specific heat, high latent heat of vaporization. That's important, of course, when water is evaporating off leaves. Transpiration is a sort of, there's the indirect effect of cooling the leaf and the canopy. And so you need a large energy input to vaporize water. It comes from the sun. So I mentioned transpiration is important. I'm trying to wake some people up here or put them to sleep. Water has a high surface tension, so it's important for cohesive and the cohesive properties of water, and it adheres to different surfaces as well. Again, due to the hydrogen bonding, it holds together very well, and the strong surface tension keeps it a liquid at the same temperature as I mentioned. The surface tension is about 3 times higher than ethanol, and almost twice as high as phenol. So this allows water to sort of stick to itself and stick to other things inside of the plant, allows the water to be drawn up by the plant. And all of these properties, as I said, contribute to capillary rise. If you look over here, this is just a diagram of, for example, water when it's on a hydrophilic surface or a surface that really attracts water molecules, it spreads out. When it's on a hydrophobic surface, you get this really nice droplet, and water sticks to itself. The capillary rise here, you can see a comparison between water and mercury, for example. is that when you put a capillary tube in this little basin of water, the water sticks to itself and it sticks to the walls of the capillary tube and it gets drawn up naturally. There's no transpiration or anything going on. Whereas mercury has a very low adhesive properties, and it actually is pushed down by the capillary tube there. So just an example. And of course, water has high tensile strength, so it's really hard to compress it, and it's really hard to pull it apart. And all of this is tied to how easily it is, or it's tied to how water is easily taken up by plants, grapevines, and any other plants, and drawn through the xylem. So transport processes of water, again, just a review here, going back to basic plant physiology here. Diffusion is probably one of the most important processes. Just in general here, you can see we've got 3 beakers. If you imagine that you've got a concentration of 2 solutions that's the same, but they're separated somehow when you first put them in the beaker, over time they'll mix, and then eventually they'll be evenly distributed. And so diffusion is the driving force. It's a difference in concentration between solutes in a solution, and it's a passive process. So there's no energy involved. It just happens naturally when you have 2 solutions with different concentrations, and it's very, very efficient at small distances, like at the cellular level, moving stuff within the cell or between cells. But as the distance increases, it becomes very non-efficient. So if you would imagine the time for a molecule of oxygen, for example, to go 1 micrometer is 250 microseconds. The time for it to go 1 millimeter is 4 minutes, and the time for it to go 1 centimeter is 7 hours. So diffusion, again, really, really good at short distances, like when we talk about water molecules leaving, going into different parts of the leaf or leaving the stomates. But in terms of moving molecules long distances, you're going to need some more energy. Osmosis is basically diffusion, but there's a cell membrane involved. So it's a semi-permeable membrane. This is important, for all of biology, basically. And the idea is kind of the same here. You've got high concentration of solutes on one side of the membrane, low concentration of solutes on the other side of the membrane. And in this case, the water is going to move from here to here to sort of balance out the concentrations. And then there's bulk or mass flow. This is sort of when you put water on the soil, and you got a wetting front that moves through the soil, and it's going to move everything with it. And the same thing can happen in a plant, for example, in the phloem. When what we have is the cells are loading the phloem with sugar, water rushes in because there's a high concentration of solutes. That water rushes in, creates a pressure, and the water moves bulk flow down through into the whatever sinks, shoot tips, root tips, fruit. You guys with me still? All right, I'm going to talk about vines. So soil plant atmosphere continuum. You've probably all heard this. Water potential, water flows downhill. Water potential is sort of, we talk about energy states of water, chemical potential energy. Pure water has a water potential of 0. And it has the highest potential energy to do work. And it always is going to flow from high water potential down a gradient to lower water potential as that high energy is released, and it's going to continue to flow downhill to lowest and lowest water potential. Now this is important because the soil always has the soil solution has the highest water potential. The root has a slightly lower water potential. The stem has a slightly lower water potential. The leaf even lower than that, and the atmosphere even lower than that. So this drives the entire movement of water from the soil through the plant into the atmosphere. And the stomates are sort of the regulators of all of this, and so the vines are directly connected to the atmosphere through these tiny pores. And they're really responsible for controlling all of the water loss and the uptake of CO2. So the stomates are going to open gradually in the morning as the sun rises. At about 1/3 of full sunlight, sort of mid-morning, they're typically fully open under well-watered conditions. And then when the sun sets in the evening, they close. And CO2 doesn't diffuse quite as easily into the stomates as water diffuses out, but you can see here kind of what I was talking about. This water's coming from the xylem, which came from the root, came from the soil. That's going to go through here, and it's going to leave through the stomate here. This is sort of the atmosphere on this side, the leaf on this side. And the one thing that I'll mention here as far as the stomates are concerned is that the stomates are wide open when the plant is at full turgor. So when the plant when it's well watered, when the leaf is well watered, when the water potential is really high or not very negative, the stomates are going to be wide open. And as that leaf starts to dry out and the plant starts to wilt a little bit, the stomates start to close. So the guard cells here, which control the opening and the closing of the stomates, basically what happens is when they're at full turgor, the guard cells are inflated and they're open in the stomate. And when the leaf starts to dry out, they lose turgor, the water pressure goes down in the guard cells, and it closes. And so that's kind of how the stomates work. In the xylem, this is a really nice sort of 3D X-ray of a grapevine xylem. And these are just giant pipes. Grapevines are lianas. They're vines. They're highly specialized to move water, much more so than trees. Lianas, grapevines, and other similar types of vines, their xylem vessels are about 4 to 10 times larger than those of trees. So what grapevines are really good at is making leaves, making fruit, and they don't invest a lot in wood. So trees invest a lot in wood production for stability. I had a professor when I was at Davis say grapevines are structural parasites. So they don't make a lot of wood. They're really good at making pipes for moving water, but they're not really good at making wood for standing up on themselves. So they need to stand up on something else, either a stake that you put in the ground or a tree, by the river. But they're highly specialized for moving water, and their xylem vessels are very, very large. One thing I'll point out here, just a sort of a point of interest, these vessels are very large, and you can see these little kind of ribbed indentations here. These are all pit membranes, and these are just the interconnecting sort of membranes between the vessels. So if you kind of look through this vessel on the other side, there's another vessel, and it's connected with these pit membranes. And the water can move not only within a vessel up and down very easily, but also move in between vessels through those membranes. And so there's a gradient from the soil solution into the root as well. So here we have the soil particles. They're kind of holding onto the water. And in different places, the water is much more easily accessible by the root. We've got a root hair here. And in the root, we have a lower water potential than in the soil. So the water is going to rush into the root, and then it's going to move through the root into the xylem. Now, I don't have a picture here, but there's two pathways that water can get from the root, from the soil solution through the root into the xylem. There's the apoplastic way, which is sort of through the network of cell walls. Sort of like if you put water on your t-shirt and it gets soaked up, that's kind of that capillary action. And there's also the symplastic way, which is directly through the cell membrane. And all plant roots, for the most part, have, and it's not depicted here, but if you would imagine sort of like a belt around the root, and it's a waxy belt that forces the water, that before it gets to that belt, it's going to move either through the cell membranes or through the cell wall. But when it gets to that belt, it has to go through the cell membrane. It's called the Casparian strip, and a lot of that transport through that membrane is regulated by aquaporin. So I'm not going to talk about any of that, but just know that the plant I like to always think about it as almost like the plant's built-in filter. Like the root has a lot of ways to take up water, and then at some point it's got a little filter built in that has to filter everything that's going through the root through the membrane. And how permeable that filter is would sort of dictate how easily the water is going to move through the root. So when you have the whole picture here, you can see that you have a low water or a high water potential in the soil here. It's going to move up through the roots. The water potential is going down, 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 and the atmosphere has a very, very low water potential. So that's kind of the sort of system at large. So daily and seasonal vine water relations. Here's some of Larry's data that I will show quite a bit of his data, but such is life as a former graduate student of his. So here we have just the diurnal time course across the x-axis. water, grams of water per 2-hour period on the vertical axis here. And you can see there's two curves here. There's a transpiration curve and there's an absorption curve. And when you think about that whole system from the soil solution out to the atmosphere, it's not free of resistance. There's resistance along this pathway. So the water doesn't move very easily. And so what you have is the transpiration increases and the water that's in the leaves is going to leave the leaves. That's got to get replaced by the water from the xylem. The xylem water's got to get replaced by the water from the root. And the root water's got to get replaced by the water in the soil. But there's a time delay due to the resistance in this pathway. And so what you see is that the pattern of transpiration doesn't line up exactly with the pattern of absorption from the soil. And when you look at a diurnal time course of water potential, this is, again, Larry's data looking at leaf, stem, and cluster water potentials. So the top three curves with the circles, those are very well watered. And the bottom three curves with the squares are watered at 40% of ET. And you can see that they, in general, have the same pattern, where they start off pretty high, they go down to sort of a low level in the middle of the day, and then they recover at the end of the day. And you can see that both the leaf, the stem, and the cluster all follow the same pattern. And I always like this cluster water potential because it's fun to imagine shoving a cluster into a pressure bomb. Do you have to cut the cluster like in little pieces when you do that, Larry? Yeah. So daily minimum between 1 and 3 p.m. Solar noon is 1:15 or 1:10. You can look at that. There's a cool app called Sunseeker that my buddy told me about, and you can look at the angle of the sun, and it tells you exactly when the sun is at the highest angle, and it's, at least where I live in Medford, it's like 1:15. I'm assuming it's similar here. Over the course of the season, if you keep vines well watered, the water potential will stay pretty high. If you deficit irrigate them, the water potential is slowly going to go down because the vine's taking up water from the soil and you're not replacing all of that water that it's using. So here what we have, again, midday leaf water potential on the vertical axis. Day of the year on the bottom. We wait until the water potential gets to some threshold, then we turn the water on here at this arrow. And we're watering at 120% of ET here, and the water potential is pretty much staying fairly high. If you're deficit irrigating at 80%, it's slowly going to go down, and if you're deficit irrigating at 40%, it's going to go down even more. And the idea here is just we're not replacing the water that the vine's using in the soil, so there's less soil water available, and it's harder for the vine to pull the water out of the soil, and the water potential is lower. Oh, the font is different. So here's some data looking at how cultivars are different. This is sort of a known thing. We noticed this in the vineyard, that cultivars respond differently to water stress. These cultivars were grown on the same rootstock, and we made a lot of different measurements of stomatal conductance and leaf water potential on the same leaves. So we measured the stomatal conductance and bomb the leaf. And when you look at the data over the whole season, we have 3 cultivars here, Tannat, Cab Sauv, and Petit Verdot. And so the midday leaf water potential is now on the x-axis. So we're going from wet to dry, from right to left, this way. And the stomates are really open up here and really closed down here. And you can see that, for example, Tannat has this really sort of a very strong sort of sigmoidal curve, and where a cultivar like Petit Verdot doesn't really. And so a couple things is that the maximum level of stomatal conductance is actually pretty similar between 400 and 500 millimoles per meter squared per second. So the maximum stomatal conductance is not that different among these 3 cultivars. But their response to drying is quite different. And so I pointed out these different leaf water potential levels. These levels are sort of when we assume that the stomates are going to start to close. So we're going to say, all right, based on these curves, these levels of leaf water potential indicate when the stomates are 95% open, and from then they're going to start to close. And you can see for Petit Verdot, they're starting to close at minus 0.88 megapascals or 8.8 bars. Cab is a little bit later, maybe 1 bar later. And Tannat's almost at 13 bars before the stomates start to close. So this is important when you're making measurements to try and figure out how to determine what's the best irrigation management strategy. The other thing I'll point out is that once they do start to close, that the stomates of Tannat are closing super fast relative to Petit Verdot. So, okay, Petit Verdot starts to close the stomates at a higher water potential. It's wetter when it starts to close the stomates, but it kind of closes them in a fairly relaxed way. Whereas Tannat almost has this like, we're open, we're not open. And so we actually looked at 17 different varieties. And so on the x-axis, what I've plotted here are these these data points where we say, all right, the stomates are now starting to close. And what is that water potential when the stomates start to close for all these different varieties? And how fast do they close? And so you can see that the varieties are all sort of distributed fairly evenly here. The later or the drier, the more dry the water potential is, the lower the water potential is it takes to get those stomates to start to close, the faster they're going to close. And these varieties are all just kind of different. And a lot of people talk about, isohydric, anisohydric, this and that. And the one thing I want to point out about here is, do you guys see any sort of like distinct grouping? Like, do you see like, some varieties are like one way and some, they're not. They're kind of all spread out. And the one other thing I want to point out is that I've circled 2 data points here. These are Grenache and Syrah, sort of the stereotype of these different behaviors of stomatal conductance as a function of water status. And what I'll point out is that on the vertical axis, they're about the same. They're closing the stomate. Once they start to close the stomates, they're closing them about at the same rate, which was interesting to us. What you do see here is sort of what you might find in the field is that Grenache is going to wait a little bit longer before it starts to close the stomates. And Syrah is going to start closing them at a lot higher of a water potential. Or actually, maybe that's opposite what you think in the field. Maybe a lot of people think that Syrah is going to close the stomates really fast, and Grenache doesn't. But in any case, there's no distinct grouping here between these varieties. They're all kind of all over the place. They're spread out. The later a certain variety waits to close the stomates, the faster it's going to close them. These are all grown on the same rootstock. Rootstocks, of course, have an effect, or maybe they don't. So here's some data from Larry, 2010. And we've got 5 different rootstocks, and I think this was cab. And basically we have different levels of irrigation here on the x-axis, midday leaf water potential on the y-axis. So of course, okay, you don't water the grapes, water potential is really low. You water them a lot, water potential is really high. But you don't really see a clear distinction between these rootstocks. Yeah, maybe a little bit here and there, but typically you water them less and they're going to be drier. And so the degree to which the rootstock changes the water status of the vine is likely to be a lot less than the degree to which the scion cultivar is affecting what's going on. So some factors affecting grapevine water use. So here's 2 graphs right next to each other. The one on the left is solar radiation and ambient temperature diurnal time course. So we've got time of day on the x-axis. Time of day on the x-axis for both figures. On the left, we got solar radiation on the left, so it's really, really sunny. Here, ambient temperature, really, really hot. And you can see they're sort of shifted in time. Something what you might expect. Of course, the sun comes up. You know, we're still in the solar system here. Everything's pretty straightforward. Sun comes up, and then it goes down. Light increases evenly and then decreases. Solar noon, kind of right around here. But temperature isn't the same. Temperature takes a little bit to heat up. And max temp, sort of like 4:30. And then it cools off. You can see for the most part that the vine water use kind of follows the net radiation. So the sunlight is driving the evapotranspiration primarily. The one thing I will point out is that you see this little kind of shoulder over here off to the side, and that's because it is quite a bit hotter and quite a bit drier in the second half of the day. But overall, the take-home message is that typically the sun is driving water use. And that's going to be important here in a second when I have some other field data to show you. The other thing that affects water use, we talk about crop coefficients. Larry's going to talk a little bit more about crop coefficients. But of course, the crop coefficient depends on your row spacing. If you've got more vineyard surface area covered in more rows, which have more plants with more leaves, the water use on a field scale is going to be a lot higher, right? If you've got 1 row in an acre, you're going to have some amount of water use. And then when you put 2 rows, I bet the water use will double. And so all of this is saying that the crop coefficients should take into account row spacing, because it is going to make a difference in terms of when you're scheduling irrigation. I won't talk too much more about that because that's Larry's next talk. And of course it depends on trellis type. The trellis type sort of determines how much of that canopy is going to be displayed. How big of a solar panel are you putting out there? If you're on a VSP and you're like this, and the sun is directly overhead, do you think that that canopy is intercepting a lot of that sunlight? No. If you're, jeez, man. If you're on quad cordons and you got a split canopy and you got a big bushy canopy and you're intercepting a lot of light, the water use is going to be a lot higher, right? And so here you can see the difference. VSP is way down here in terms of how the crop coefficient develops over time. You got some intermediates here, and then you got the quad cordons here at the top. At some point they all max out because the canopy closes up and that's it and stops growing for the most part. Vine water use is also a function of soil water depletion very strongly. This is just like, you can think of this as crop coefficient, but it's ETC, crop water use versus reference water use. And it's a linear relationship. As soil water content goes down, this is averaged across multiple depths as just straight up. As there's less water in the soil, that's going to change how much water use the vine, or how much water the vine uses. However, some interesting data, again from Larry. This is looking at how does the environment influence the water status of a vine if the vine is already wet or if it's already dry. So if you look at the curve on the top here, and here we've got VPD, vapor pressure deficit, sort of like a measure of how dry is the air on the horizontal axis. On the vertical axis, we've got leaf water potential. And when the vines are well watered, so soil water is not limiting. We're at field capacity, or whatever you want to call it. The environment is the primary driver of what's going to give you that lower or higher water potential. So if it's really dry, it's like 110 degrees and relative humidity of 10%, you know, like in Fresno every day, the water potential is, and the field is wet, the water potential is going to be low, period. The vine can't do anything about that. The environment is driving that. Because it's really, the vine is screaming, it's transpiring a lot of water's coming out of the ground, it's going through the vine, there's still a lot of resistance along that flow pathway, and the water potential is going to be really, really low. Similarly, if you're on the Sonoma coast or something like that, and you're at field capacity, and it's not that dry, the water potential is going to be pretty high. Both vines are wet, but this is just an illustration of how the environment's controlling that. However, if you have a dry condition, and soil water is limiting, the environment's really not going to do a whole lot. You can see this curve is pretty flat. So all that's saying is that at this level, when the vine's dry, the stomates are really kind of controlling what's going on in the grapevine. And typically, this is sort of like rule of thumb, about 12 bars, minus 1.2 megapascals. That's really when the stomates kind of take over. Of course, I showed you different varieties are different, so keep that in mind. But rule of thumb is 12 bars if you want something to take home. Vine water use over the course of a whole season. I've got 2 seasons of data here from a research site I have outside of, I think this is outside of Grants Pass in Southern Oregon. This is daily ETC, estimated ETC in inches per day. And so we've got 2 years here. We've got the black lines kind of bumping around. This is every single day, estimated crop water use. The blue line is just a smooth kind of weekly average. And you can see it increases to a certain point, kind of maxes out, and then goes down over time. And you can see there's variability every single day. When it's cloudy, there's less water use. When it's really sunny, there's more water use. We had right over here, we had a little bit of a cloudy kind of rainy event here for several days, about maybe a week, 10 days actually. And the water use went down quite a bit. And then when the sun came out again, it went up again. Now, the absolute values on the y-axis here are sort of irrelevant for our discussion, only from the standpoint of this is just a function of the evaporative demand at this particular site. The trellising, this is a VSP, 8-foot rows. The environment is going to be different there than it would be in Central California, for example. So don't pay too much attention to that, but you can see the general pattern, variability during the day and pattern over the season. I did want to poll the audience. We had these kind of depressions here late in the season in 2017 and in the middle of the season in 2018. Can anyone venture to guess what happened? Smoke. Very good. So we had, in 2017, we had a thunderstorm sort of early August. It was one of these sort of dips here probably, and then wildfire smoke filled the valley. And you can see here that ETO and ETC crashed because the sun blocked out. And then can anyone guess what this super low number is on August 21st of 2017? The eclipse. So look at that. Look at how low ETC was. Pretty cool. So it was smoky and there was an eclipse. I mean, talk about water use efficiency. I kid, though. And then in 2018, we had a very large smoke event very early in Southern Oregon. We had a lightning storm on July 12th or something, and it was something like 300, 400 lightning strikes or something like that. And we had a bunch of fires, and you can see this kind of flat top action of crop water use. The totals, these are just based on phenology that I put these numbers in. So bud break to bloom, about 10% average of your total seasonal water use. Bloom to veraison, about 50%. Veraison to harvest, about 30%. And then after harvest to leaf fall, about 10 to 15% total seasonal water use. Again, I've got the numbers in inches and then in gallons per vine in parentheses. Those are sort of irrelevant on a, those are kind of absolute, but the relative values are what's important. How long do I have? Till 9:15. All right, nice. Plant responses to water stress. This is a figure that's older than I am. It's a very sort of a famous figure, I would say. It sort of depicts general plant response to water stress. So the stress starts, and the plant is dead, over here on the right, and sort of what happens. So the first thing that happens, I like to call it some sort of growth adjustment. Canopy doesn't grow quite as much. There's an increase in carbon allocation to grow more roots rather than more shoots, and those happen almost right away. And so growth is one of the most sensitive, if not the most sensitive, response to water stress. And I'll just say that these sort of weird shapes is just kind of like the intensity of the response. So when it's really narrow, it's not very intense, and as it expands on the figure is just an idea of like, well, it's really strong. After there's some sort of adjustment of growth and carbon allocation, I call this drought management. So there might be some osmotic adjustment, more solutes being sent to the leaf to keep the stomates open or to close or not to close them. Stomates start to close at this dashed line. Drought escape is kind of the last thing that plants typically do. They'll turn the leaves away from the sun. They'll put them down. The leaves will fall off. As far as the plant is concerned, it's doing exactly what it's supposed to be doing. As far as you're concerned, you're freaking out because the leaves are falling off. But the plant is doing exactly what it's genetically programmed to do. Rating tendrils or looking at tendrils in the field is a common thing that we do to sort of say, I don't need a water, they're fine, look at those tendrils. I like to grab the shoot tip and just hold the end of the shoot tip and you can see the tendrils sticking out. It's easier than, you can look at them, but then they're kind of floppy. The good Dr. Mark Greenspan has provided the internet with this image that I stole from him. Is he here? No. Bummer. He what? Oh, good. I was going to say, so Mark likes to say I can look at the vine and tell you what the water potential is. So I was going to sort of troll him and put water potentials here, but I didn't. Anyways, looking at the shoot tips is a good way of sort of roughly estimating what's going on with the vine. However, even though I say that growth is the most sensitive thing, the problem is that a lot of the times when you see something in the vine, see a change in the shoot tip or the tendril length or whatever, it oftentimes might be too late. And to further complicate all of this, here's some old data from Hans Schultz and Mark Matthews. This is a plant water potential on the horizontal axis and growth rate on the vertical axis. And there's 3 curves here showing different responses of tendrils, leaves, and internodes to water stress. So we're sitting here saying, well, we're going to look at the tendrils, but we're also kind of, in our vision, we're seeing leaves and we're seeing shoots, and every single one of these organs has a different response. Either a different sensitivity. For example, the growth is most sensitive in tendrils, so we're kind of on the right idea. Leaves are most sensitive after that, and then internode elongation is the most sensitive after that. But the growth stops actually for leaves at the highest water potential. So at 10 bars, the leaves stop growing, but the tendrils keep growing until 13 bars, even though they're the most sensitive. So what are we looking at? I mean, it's a much more complex picture. Leaf angle and sun avoidance. This is some data from my PhD, which, Larry, I'll be publishing this, don't worry. This is like the fanciest way to depict plant wilting you'll ever see. All this says is that when you move from wet plants on the right to dry plants on the left, the leaves do this. And it's statistically significant. So, but it's important because that's another sign that we say, oh, the plant's dry, the leaves are wilting, and maybe I should irrigate. However, not to throw something else in the fire, turns out that cultivars have different leaf angles just by default. So some cultivars have more erect leaves than others. So what are you looking at? Are you looking at Cab? Are you looking at Grenache? Are you looking at all these different varieties? Some of them have more erect leaves than others. So when you're looking at one variety that maybe just has floppy leaves, you're like, oh my God, I need to irrigate. But it just has floppy leaves. Or maybe there's another variety that has really erect leaves. And you're like, nah, it's fine. It turns out you really need to irrigate. Leaves, when they dry up, they fall off, and it's always the basal leaves. We looked at the number of leaves that fell off of a shoot as the plants got drier. These data points are averaged over 17 varieties in 2 years. And you can see that the leaves stay on the vines, stay on the vines, stay on the vines. And when you get to about 13, 14 bars, then they start to fall off a lot, really fast. And we were measuring vines out here that basically had not very many leaves on them. Shoots start to slow down as well. So if you have deficit irrigated vines, the shoots are shorter. Shoots are longer here in wet vines. Sort of an obvious thing. This has to do with internode elongation. And at the end of the season, if you have really dry vines, you have lower pruning weights. So all of this is just to say vegetative growth is fairly sensitive. Reproductive growth. If you think about just yield and pruning weight as 2 gross measures of reproductive growth and vegetative growth, and you have sort of a relative maximum value, and you're irrigating different percentages of ETC, pruning weight responds almost in a linear fashion. More water, more shoots, more leaves, and it just keeps going. When you irrigate and you're looking at yield, it also responds linearly up to about 60 to 70% of ET. And then it just kind of levels off. You're not getting any more fruit. That's it. You're just growing more shoots and leaves. So if you want to grow more shoots and leaves, that's fine. But you're not going to get any more crop off those vines. So take-home message here is that reproductive growth is much less sensitive to water deficits than vegetative growth. So depending on your production goals, I would say you really don't need to be irrigating at 100% of ETC unless you really want the vines to have like a lot of shade over the fruit, maybe for growing table grapes or something like that. Berry growth has 2 growth phases. There's a first growth phase, and then it stops, and then they turn soft and purple if they're red, and then they grow again, and then you pick them. We're all familiar with this, hopefully. The interesting thing about berry growth and how the fruit is connected to the vine hydraulically, before veraison, to the left of this line, this is expressed as days relative to berry coloration, You can see the berry diameter here on the vertical axis, and it's bouncing up and down. Remember those diurnal curves we saw of water potential? The berry's doing the same thing. Before veraison, the berries are expanding and contracting every day. But then as it approaches veraison and then colors up and then starts to grow again, they're not really doing that so much. There's not a lot of contraction. It's just expansion and then it sort of stops, and expansion and sort of stops, expansion and sort of stops. But if you were to measure diurnal water potentials during that time, you see the same thing. So what happens to the fruit is after veraison, it becomes more hydraulically isolated, and it's not quite as connected to the grapevine as it was before veraison. And expressed in a different way, you can see this. Here's the leaf water potential of the vine and the percent of the diameter that the berry is contracting and expanding. And I think this is Zinfandel, I'm not sure. Before veraison, there's a really strong relationship. So when the plants are really wet, this time it's really wet on the left side, they're expanding and contracting a little bit. And then when the plants dry out, before veraison, they're expanding and contracting quite a bit. After veraison, there's no relationship. You can have a wet plant, you can have a dry plant, the berry doesn't care. It's going to grow. We looked at this as well across different varieties. And so we have really dry plants on the left, really wet plants on the right. We have the berry absolute growth rate in milligrams per berry per day. The solid lines for these four varieties, Cinsault, Montepulciano, Cabernet Sauvignon, and Petit Verdot, represent the pre-veraison response. So you can see, just like in the previous figure, pre-veraison, there's a really strong relationship between how much that berry grows every single day and the water status of those vines. It's a linear function, really, really strong relationship. The varieties are different. So Cinsault has giant berries, and the absolute growth of those berries is going to be larger than the absolute growth of Petit Verdot, which has very small berries. But it's still a significant function of how wet or dry the plant is before veraison. After veraison, the relationship falls apart. The berries are doing whatever the hell they want. You can have no water on the vines, a lot of water on the vines. The berries are still going to grow to a certain degree. And then at some point late in the season, if they're still really dry, once the berries get to some level of bricks, they typically start to desiccate. You've probably all seen this as well. The point is that the fruit is much more sensitive to the water status and how you're managing irrigation before veraison, and much less sensitive after veraison. So if you're really trying to control berry growth, the chance to do it is before veraison. And after veraison, the berries are going to grow. If you messed up early and you wanted big berries at the end, you're not going to make up for it by dumping a lot of water on the vines after veraison. If you wanted small berries, cutting the water off after veraison is not going to help you out. The chance to get small berries is early, between set and veraison. That's it. Any questions? Okay.
I'm wondering on those two graphs that you showed from 17 and 18, where it showed the differences in ATC, how did you get all those big ones? Did you do a clock, or is there some kind of constant?
So this is estimated ETC. All I'm doing is looking at reference, and I have a crop coefficient. I'm not measuring anything. This is from a weather station.
What's that crop coefficient comes from?
It comes from Dr. Williams. What's that?
I mean, it's related to a certain canopy?
Yes. Yeah, it's a function of growing degree days. So we basically say, like, all right, bud break, start accumulating growing degree days. There's an equation, an empirically generated equation. And I think, Larry, you developed, at least for the VSP one, you're using water balance, right, with a neutron probe? So it's an empirically derived equation based on growing degree days.
Can you comment on rootstocks? We get a lot of questions in California that people are touting drought-resistant rootstocks. Can you differentiate between tolerance and resilience?
Oh, geez. I would ask you to define what you mean by drought tolerance.
Well, I don't know. I mean, I get these questions. I'm like stumped.
So one of the things that I noticed firsthand when I was doing a lot of my work in California, we were using these like Rupestris berlandieri rootstocks, 1103P, for example. And and we're working on valley soils, really well drained, very deep, very easy to manage as far as irrigation. You stop watering it, they get dry, you turn the water on, and they get wet again. It's like turning a light switch on and off. And it was great for scientific experimentation. When I went up to Oregon, and the soils are a little bit heavier in southern Oregon, and we're using like 10114, for example, and it was like I wouldn't water this thing, and it was just staying wet forever. Staying wet forever, staying wet forever, staying wet forever, and then all of a sudden, boom, it got super dry, and then when I turned the water on, nothing happened. And so there's definitely a genetic component and a I don't know what that means. You might argue that it's more drought tolerant because it's staying wet longer. Or maybe it's less drought tolerant because once it gets really dry and, crashes or whatever you want to call it, and then you turn the water back on, nothing happens. So it's important to quantify what you mean about that. But in terms of management, it was challenging because what's going on? One, turn the water on and off, it's like a light switch, and the other, you turn the light switch on and off, and the lights are just on, and then they go off, and then you can't turn it back on again. And so there's definitely a difference among rootstocks in terms of water uptake, and we're still trying to sort all that out, I think. And I think it's a challenge. I think anecdotally, what you experience, and I don't know what your experience is in the field, I mean, does that match like what I said about those two?
Yeah, it matches what you say, but this is coming from a sales pitch to our growers most of the time.
Yeah.
And then our growers are hitting us up with those questions.
Yeah, it's tough. I would say that just off the bat, having dealt with that firsthand, not from a management standpoint, but from an experimentation standpoint, it's really tough. It almost seems like for the rootstocks that are like, 10114, 3309, those kind of more riparian rootstocks, they kind of tick down more slowly, and I would be really careful about super intense deficit irrigation on them because they may not recover so easily if you sort of go over that cliff. So you want to stay on the good side of the cliff. The other ones tend to come back a little bit more easily. So you can really farm them pretty hard and not water them as much, but then you know that when you water them again, they'll come back. But if there's no leaves, there's no leaves. So. Yeah, George.
Alex, you have very interesting slides about the storm exposure and the new daily water potential. So, what's your comments about the drought-resistant variety or the heat-resistant varieties? Because I got all of you asking, is there any real drought-resistant or heat-resistant varieties?
Again, it sort of depends on. It depends on what you're looking at. In terms of this data set, for example, these are the ones that are keeping the stomates open under more dry conditions, more drought resistant. They're using more water. They're not conserving water. They're keeping the stomates wide open. You might make an argument that this one down here is more drought resistant because the stomates are closing fairly early and it's using less water. What would be interesting is, so one thing I'll say this, if you keep the vines wet all the time, and the maximum stomatal conductance is about the same across these varieties, then the thing that's going to drive whether or not one is using more or less water is its relative vigor and how big the canopy is and how the trellis is configured. If the maximum stomatal conductance is the same and everyone's on a VSP and you're hedging all the time and the canopy is just a curtain, there's not going to be a huge difference. It's going to be a pretty small difference. If the maximum stomatal conductance is the same but you're on a more sprawled out trellis and you've got differences in vigor that are giving you different size canopies, then you will notice a difference in water use between the varieties. In terms of manipulating the water use of the vine by drying it out or rewatering it, I don't know if we're in a position to be tricking these vines or finding ones that are more drought tolerant or anything. And ultimately, it depends on the crop. I would want to evaluate the drought tolerance or resistance of a grapevine, more on what's going on with the fruit than what's going on with the canopy. This isn't wheat. This isn't like, we watered less and got the same yield of wheat. You know, it's all about the fruit. And as I showed, the fruit is much more, in general, or much less sensitive to reductions in irrigation water than the canopy. I don't know if that answers your question, because I just threw a bunch of stuff in there.
All right.
All right.
Well, Alex will be around the rest of the day.